Okay, guys and gals, lecture capture is recording, so I'll begin. So we're going to continue an examination of sensory and motor nerves, and indeed sensory and motor systems, in particular in relation to the spinal cord. I have to confess that the spinal cord is not the most interesting part of the nervous system from the point of view of optometry. But it will allow me to introduce some really important kind of principles or concepts about how the nervous system is organised, and we'll follow on some of those next year. And it'll also allow me just to sort of introduce briefly the autonomic nervous system, which I'm sure you've heard of because Ron Douglas told me he's mentioned it to you already. And actually the autonomic nervous system, some of which is embedded in the spinal cord, is really important for optometry, and it's the subject of the next lecture, and therefore worth sticking around for. Right, so as a little reminder, I told you earlier today, you know, that the number 2 is kind of important for nervous systems, because there are 2 major parts to the nervous system: the central nervous system, comprising the brain and the spinal cord, and the peripheral nervous system, which has a whole bunch of nerves which are connected to different tissues, in fact all of the tissues of our bodies. But here's another 2. The motor component of the nervous system also has both a voluntary and an involuntary component to it, and that's the autonomic, which is the involuntary bit. You see the word autonomic sounds a little bit like automatic. It's the bit of the nervous system over which we have no voluntary control. It's doing stuff behind the scenes of which we have no sort of conscious awareness. So I just thought I'd mention a little bit about it now as a prelude to the next lecture. So what I talked about earlier today was, for example, that you have sensory receptors of different types in your skin that pick up different types of somatic sensation, and they report that information by these afferent sensory nerves to parts of the central nervous system, and that there are also motor neurons inside your central nervous system that send axons, like this mauve one down here, to innervate the voluntary or skeletal muscles, and indeed that is the voluntary component of the motor system. But the picture also shows you, or gives a little indication of an organ which is supplied by components of the autonomic nervous system. So I just thought I'd talk a little bit about it. So what this picture is intended to show is that there are a type of sensory receptor that are located in the walls of your really large arteries. I'm thinking about the aorta and the carotid artery, which is a big one that runs up through your neck. And the function of those receptors, they're called baroreceptors, is to monitor the blood pressure in these large arteries. Okay? In fact, they're a type of stretch receptor, a little bit like the sort of Ruffini endings that I mentioned that we have in our skin that signal stretch. and that we find in our joints. And the function of these baroreceptors is basically to monitor the strength of the pulse. Now, under certain circumstances, that your blood pressure, might be a little bit elevated, maybe elevated more than you'd really like. That condition is called hypertension. And under those circumstances, it might be, and the baroreceptors will signal that information to the central nervous system that your blood pressure is a bit high. And under those circumstances, you might want to lower your blood pressure a little bit. So there are motor nerves that come from particular motor neurons within the central nervous system that innervate the cardiac, that is to say heart muscle, and slow the heart muscle down, reduce its force of contraction. Therefore, the pulse pressure drops, and your blood pressure would return to equilibrium. On the other hand, it may be the case that your blood pressure is a bit too low. That's called hypotension. And actually, that's a little bit more serious, hypotension. If your blood pressure drops too low, it can make you feel quite dizzy, and in really quite severe circumstances, you can even lose consciousness. And the reason for that is that blood isn't being pumped in sufficient amounts to your brain, and under those circumstances, you can fall unconscious. So under those circumstances, other motor nerves, which innervate the muscle of the heart, might want to increase the force of contraction and therefore increase the blood pressure, and again return it to an equilibrium. And both of those mechanisms, both reducing high blood pressure and increasing low blood pressure, are mediated by different parts of the autonomic nervous system, which we'll talk about in more detail in a little while. Okay, so it turns out that there are in fact 31 pairs of nerves. By pairs, I mean you've got one on the left and one on the right, which are directly connected to your spinal cord. And as we'll see, what those nerves are doing is taking either sensory information or sending motor signals to pretty much all of your body below the neck. Fortunately, we don't need to know the names of any of those nerves. As we'll see next week, there are 12 pairs of what are known as cranial nerves, which are connected to different parts of the brain. And unfortunately, you will need to know the names of the 12 sets of cranial nerves, and you'll need to know quite a lot about them, which is why I'm going to spend 2 hours telling you about cranial nerves next week, and why we're going to do a lab tutorial in a couple of weeks' time that focuses on those particular nerves. But as you can see, there are a whole bunch of really big nerves that go in and out of your central nervous system. And those nerves, which are part of the peripheral nervous system, or PNS, are connected to the central nervous system in a kind of quite integrated process. I mean, these are the relationships between the peripheral and central nervous systems. This box down here is meant to be your brain and spinal cord, the CNS, and this stuff out here is meant to be your peripheral nervous system. And in general terms, the peripheral and central nervous systems are engaged in kind of 5 integrated processes. So, for example, as we saw earlier today, you have receptors in your body that are responsive to particular stimuli, and they're engaged in transducing those stimuli and converting the stimuli into patterns of electrical activity, which they then send via sensory or afferent nerves to the central nervous system. So they report that information to the brain or spinal cord. And the job of the neurons in the brain and spinal cord is to analyze or interpret that information. Under some circumstances, you might want to immediately do something about the stimulus that you've just received. For example, you may be in pain, you pricked your finger or something, and you might therefore want to move your hand away from the painful stimulus. And there are neurons within the central nervous system that are able to send instructions or commands via the motor or efferent nerves that innervate effector tissues in your body. Effector tissues is a general term for muscles, whether they're skeletal or smooth, and for glands, because smooth and skeletal muscles and glands produce an effect, you see, so they're effectors. In other words, they produce a response. So, that's the kind of overview. Now here's a little bit of detail about the organisation of the spinal cord. It is, as I've said, part of the central nervous system. It's enclosed by the bones of your vertebral column, in other words, your backbones. It's quite long, anything from 1/2 to 3/4 of a metre in length. That depends a little bit on how tall you are. And it extends from the very base of your brain down as far as the first lumbar vertebral or lumbar backbone, which is about here. You've got some bones down here at the base of your bum, inside which the spinal cord is not found. The spinal cord is bilaterally symmetrical. It has a left and a right side, and from an anatomical and functional point of view, those sides pretty much do the same thing. Okay? So they look pretty much the same. And the spinal cord is connected to all of our body tissues, skin, muscles, and indeed organs inside our bodies by spinal nerves, which pass between a little tiny gap between the vertebral bones. And the spinal cord is involved in all sensory and motor functions pretty much for the entire body below the neck. As we'll see, the spinal cord comprises 29 separate segments. When I say separate, I don't mean they're floating. They're actually connected to each other, but there are 25 of these bits of tissue that are stacked on top of each other, which we'll see have a very similar structure and function. And overall, there is a kind of top-to-bottom functional map of the adjoining body parts. And maps are really important from a conceptual point of view, and so I'll tell you a little bit more about them when we get to them. Okay, so look, this is an MRI scan, magnetic resonance imaging scan, through somebody's head. This is their brain here, which is a really exciting part of your body. This is the very lowest part of the brain. It's called the brain stem. And you can see that it's connected here to the spinal cord. This is the first part of the spinal cord here running through through the neck. This structure over here, by the way, is the tongue. This massive thing here. Okay? Your tongues are big. This is not unusual. Tongues are really big. So this is the tongue. These things here are a couple of the vertebral bones, and you can see some little spaces in between them. The vertebral bones in the spinal cord are actually held apart from each other by little discs of cartilage. They're called cartilaginous or intercalated discs. And the deal is that the spinal nerves run in and out of the spinal cord to your body through those little spaces between the cartilaginous discs. That's what normally happens. Just as a matter of interest, in some pathologies, those discs can collapse. And what happens when the discs collapse is that the nerves passing through the space gets crushed by that. And as a consequence of that, you get pain and loss of function associated with the nerve that's been trapped. I mean, a real classic nerve that gets trapped is quite a big one that comes out of the lower part of your spinal cord. It's called the sciatic nerve. Have you heard of a condition called sciatica? This is where the sciatic nerve gets crushed as it's leaving the vertebral bones. And under those circumstances, you get pain in your legs and often loss of function from your legs as well. But under normal circumstances, those discs keep the space nice and open. Okay, so the spinal cord, as I've mentioned, has these 29 separate segments. And those segments each mediate sensory and motor functions related to different body parts in a kind of top-down fashion. So the top part of your spinal cord is called the cervical. That's the bit which is in your neck. The next part is called the thoracic. That's the bit which is in your trunk. The next part is the lumbar, which is just above your pelvis. And then there's a bottom part, which is at the lower part of your pelvis, and that's called the sacral. There are 8 cervical segments of the spinal cord, 12 thoracic, 5 lumbar, and 4 sacral. Okay? 8, 12, and 5 and 4 makes 29 in total. So those are the 29 segments of the spinal cord. In general terms, the cervical part of the spinal cord is concerned with sensory and motor functions coming from the neck and from your upper limbs, including your hands. The thoracic part of the spinal cord is mainly concerned with sensory and motor functions from the regions of your trunk. The lumbar part of the spinal cord from the front part of your pelvis and your lower limb down as far as your feet. And the sacral part of the spinal cord, which is at the bottom, is concerned mainly with the back of your pelvis and the back of your legs, and including your feet themselves, okay? Including the soles of the feet. As we'll see, each of the 29 segments has a very similar structural or common structural plan. They're organised in a very similar way, although there are some subtle regional differences between segments based on slight differences in their function related to their body parts. Right. I wonder why that acquired a box around it. Okay, so the first thing to tell you is a little bit more about those spinal nerves and how they get in and out from the peripheral tissues into the spinal cord through those gaps in the intercalated, in the discs. So each of the spinal cord segments has a spinal nerve, one on the left and one on the right. And when those spinal nerves get through the gaps in the cartilage, they split into two roots. Okay? One at the back, which is called the dorsal root, and one at the front, which is called the ventral root. The dorsal root is a source of sensory input to the spinal cord. As we'll see, receptors that you have out in your peripheral tissues that are involved in somatic sensation are attached to axons which belong to a particular type of cell which are called dorsal root ganglion cells, and the other part of their axon sends sensory information into neurons within your spinal cord in the dorsal part. The ventral root sends outputs from the spinal cord, specifically motor outputs that are either voluntary and go to the skeletal muscles in your body, or that are involuntary and that go to smooth muscles or glands or organs in your body. And that's part of the autonomic nervous system. So here's a picture of a kind of, of a standard spinal cord segment showing these nerves and roots. So this is the tissue of the spinal cord here. This kind of H-shaped arrangement is the grey matter of the spinal cord. It's where you find the spinal cord neuron cell bodies and their dendrites. And the white stuff around the outside are white matter pathways, axon pathways, taking sensory information either up to the brain or motor signals down from the brain. Okay? So that's the sort of general structure. The grey matter here with the neuron cell bodies and their dendrites is on the inside, and the white matter is around the outside. Here on the left you can see, here is a spinal nerve. This is outside of the backbone, but here it's entered into the space on the inside of your backbone. It's now split into this, the dorsal root going in this direction, and here the ventral root going in this direction. Let me just explain this dorsal ventral thing. Dorsal means back, doesn't it? I mean, as in a dorsal fin of a shark. And ventral means front. And this nomenclature comes from the study of quadrupeds, you see? Because if you walk on all fours, this part of your back would be dorsal, and this part would be ventral. But because we're bipeds, what is dorsal for a quadruped is posterior for us, and ventral becomes anterior. Okay? So this terminology is just boring anatomy. Okay? Dorsal means the back or posterior, and ventral means the front or anterior of the spinal cord. Over here we see a little bit more detail about what's travelling in the roots. So look, here we've got a sensory receptor out here in the skin. Anybody recognise what that sensory receptor is? Thank you. Who did that? Paxinian corpuscle. Yeah, that's the one that responds to deep pressure, and I mentioned in a lecture earlier today. So here's a Paxinian corpuscle. This red thing is its axon travelling in a peripheral nerve and entering here through the spinal nerve and then travelling into the dorsal root. This axon belongs to the cell body here of a dorsal root ganglion cell. The word ganglion means knot, K-N-O-T, as in a knot. It's a collection, a knot of cell bodies that you find encapsulated by a bag of connective tissue outside of the actual spinal cord. So here's the axon of this sensory receptor belonging to this dorsal root ganglion cell, and here's the other part of its axon now travelling through the dorsal root and making connection with neurons here in the spinal cord. By contrast, this is the ventral part of the spinal cord. This contains motor neurons who send their axons out here via the ventral root, and those axons are part of the motor output, in this particular case, supplying skeletal muscles. And all 29 segments of the spinal cord look pretty much like this. Now, I mentioned before that there are a variety of different somatic sensory receptors that we have in the skin of our bodies, and all of these types of sensory receptors belong to different dorsal root ganglion cells. I'm not going to go through the whole lot in detail again, but just to remind you, there are some dorsal root ganglion cells whose job it is to signal pain information to the spinal cord. But they have free nerve endings on the end. There are some that are involved in thermal or temperature sensation, which are also free nerve endings. And then there are a whole bunch of mechanoreceptors, like Merkel's discs that signal texture, Meissners and Paxillian corpuscles that signal light and deep pressure, and these Ruffini endings that you have in your skin that signal stretch. So those are the types of sensory input that travel into the dorsal part of the spinal cord. Okay, so to continue the exploration of the spinal cord and its common structural plan, I've mentioned already that the inside or inner part of the spinal cord is the grey matter, which by definition is where you find the spinal neuron cell bodies and their dendrites, and the outer part of the spinal cord is white matter, which by definition is where you find the axon pathways, which are also sometimes called tracts, a little bit like railway tracks. Why are the axon pathways white? Because the axons within your spinal cord have myelin around them. You know, Ron Douglas told you about myelin sheaths. Myelin is the membrane of a particular type of glial cell that it wraps repeatedly around axons in order to insulate them in little segments. And the function of the myelin is to speed the velocity of impulse conduction in the axon and to make sure it happens faithfully, because if you have no myelin, impulse conduction fails. Okay? That's the function of the myelin. And all of the axons within the spinal cord white matter are myelinated to a greater or lesser extent. Because those axons are in the central nervous system, what is the glial cell that myelinates them? Somebody wanted to say Schwann cell. It's oligodendrocytes. Well done, ma'am. Schwann cells myelinate axons in the peripheral nervous system, but once you get in the central nervous system, it is oligodendrocytes that myelinate axons here in the spinal cord. See, that was a principle or a kind of general concept. Right, so let's have a little talk further about the inner grey matter. It's basically kind of H-shaped, as I showed you before, with three pairs of horns. By pairs, I mean one on the left and one on the right. There's a thing called a dorsal horn, and this contains spinal neurons that are concerned with somatic sensation, with touch, thermal pain, and kinesthetic or proprioceptive sense. There is a ventral horn, which contains motor neurons who send axons out to innervate skeletal muscles. So that's the output of the voluntary system. And then finally there's a lateral horn that sticks on the side that contains motor neurons that are part of the autonomic or involuntary component of the nervous system that send their outputs to either smooth muscle, glands or organs. As we'll see later, towards the end, the white matter pathways are either somatic sensory, they're either sending somatic sensory information up to the brain for further processing, or they're receiving motor instructions from regions of the brain which are sent to the ventral or lateral horn motor neurons to do their particular jobs. So this is a cross-section through a typical spinal cord segment. This is the dorsal or posterior side of the segment, and this is the ventral or anterior side. And this is the H-shaped grey matter that you find in the middle of the spinal cord. These Roman numerals here that you see indicate that the grey matter of the spinal cord, where the cell bodies are divided into 9 separate layers. Okay? So there are 9 separate layers of cell bodies within your spinal cord, and by convention they are numbered from 1 to 9 in Roman numerals. We don't need to know loads about what's in each of those cell layers, but simply that the cells are different and they're doing slightly different jobs. But overall, the important thing is that layers 1 to 6 here comprise the dorsal horn of the spinal cord, and all of the neurons in layers 1 to 6 are concerned with somatic sensation. Okay? Things like touch and pain and thermal sensation from different parts of your body. Layers 8 and 9 are the main occupants down here of the ventral horn of the spinal cord, and layers 8 and 9 contain motor neurons that innervate skeletal muscles. So those are the voluntary motor neurons of the spinal cord. And here's sticking out at the edge, this pointy bit here, which I've labelled the lateral horn over here. That's in layer 7 of the spinal cord. And the lateral horn contains the motor neuron cell bodies, which are the outflow for the autonomic nervous system that comes out of the spinal cord. Okay? So dorsal horn, 1 to 6, sensory, somatic sensory. Ventral horn, 8 to 9, voluntary motor. Lateral horn, 7, autonomic motor. Okay, so as we're about to see, each of the segments that I've talked about, all 29, receives sensory input and sends motor output to a particular part of the body surface or to our body tissues, organised in a kind of strip-like fashion in sequence from the very top cervical to the very bottom sacral parts of the cord, which kind of maps the body in sequence from the neck to the foot with some sort of minor distortions that we will look at. The adjoining segments that are mapping sensory information are called dermatomes, and those that are associated with the motor strips are called myotomes. And they form quite detailed sensory and motor maps of the body tissues, which I'm going to come to in just a moment. So look, here is a picture of the brain, and here's the spinal cord, sort of colour-coded here to show the cervical, thoracic, lumbar and sacral regions, and to remind you how many segments are in each. So 8 segments in the cervical cord in this kind of pinky colour, 12 in the thoracic cord in this royal blue, 5 in the lumbar cord in green, and 4 in the sacral cord in what? Light blue, cyan, whatever you like, okay? And this is just to emphasise the fact that the overall structure of the segments at each of these levels follows the common structural plan that I was talking about. Look, you know, here we see at a level of the cervical cord the H-shaped grey matter on the inside and the white matter around the outside. Same thing in the thoracic cord, same thing in the sacral cord. Okay? So it's a common sort of organisation. There is, however, in this picture, a slight difference between different parts of the spinal cord, which are referred to here as these enlargements. You see that the spinal cord seems to be a bit swollen here in the cervical region, and it seems to be a bit swollen here in the lumbar region. And those are called the cervical and lumbar enlargements. And the reason they're there is that these regions of the cervical enlargements, for example, are specifically associated with sensation of movement of your hands. Okay? And you have lots and lots of sensory receptors and lots of muscles in your hand. Okay? Lots of them. And therefore, the sensory input and motor output from this region of the cervical cord is bigger than elsewhere in the cord. Similarly, this region of the lumbar cord is mainly concerned with innervating your foot. Okay? Lots and lots of sensory receptors in your foot, lots of muscles, and again, for that reason, there are more neurons in that region of the spinal cord associated with both sensation and motor control of that body part. It's a general principle. You know, the most interesting, important parts of your body tend to have more neurons devoted to sensation from them or motor control over them. Okay? So hands are really important, feet are important, and therefore more neurons associated with sensation or motor control of them. So that was a kind of concept or principle. This is a picture of a person that shows the different parts of the body that receive either motor innervation, that would be called the motor map, or sensory innervation, that's the sensory map. In fact, it's the sensory map that we're looking at from these different parts of the spinal cord. You see, the colour coding is the same. There's the yucky pink, the royal blue, the green, and the cyan. And these are the body parts associated with the cervical cord. You see the upper part of the cord, cervical, is associated with the upper part of the body, the neck, and the upper limb and hand. The thoracic with the trunk, the lumbar in bright green here with all this part of the body, and the sacral part of the cord, the very bottom part, here with the backs of the legs, soles of the feet, and actually also the genitalia as well. So as you saw, what I'm trying to get across here is that there is a kind of organisation to the spinal cord. The top part is concerned with the upper part of the body, the middle part to the middle part, and the lower part to the lower part. So there is a map, essentially, of the body tissues within the tissues of the spinal cord. But it's actually a bit more than that. It's really quite detailed. I'm pointing to this strip here. Can you see there's kind of like a ring in bluish here? That says, I think it says T6. See that thing there? It says T6. It is basically a strip of tissues running in a ring around your trunk like this. And what this means is that all the sensory receptors and all the muscles that are in this strip of tissue are directly connected to thoracic spinal cord level T5. Okay? Oh, sorry, T6. The one below it says T7. That's a strip of tissue directly below T6. You see? And all the tissues and muscles in this region directly below it are directly linked to spinal cord level thoracic T7, which is directly below T6. And then it goes to T8 and T9 and so on. Okay? So it's actually really quite a detailed map of the body parts within the spinal cord. And nervous systems, brains and spinal cords go in for this all the time. This is a general concept or principle about how the nervous system is organised. The nervous system likes order. It doesn't like chaos or randomness. Okay? It likes maps of body parts because they're put together in a sequence, in order. It doesn't like randomness at all. It does occasionally go in for a bit of randomness. I mean, here's a little bit of a distortion in the body map. You see this, the person in this particular image is lucky enough to have one of these things. And what this says is S4. S4 is the fourth level of the sacral cord. That's the lowest level of your spinal cord. There isn't an S5, okay? There is nothing below S4. And what this is saying is that sensory input from your naughty bits, it's not just one of these, but also from female naughty bits as well, ends up at the very bottom of your spinal cord amongst neurons that are receiving information from that body part, and just above that are the soles of your feet. Okay? Now, I don't know about the other guys in the room, but for me that's, you know, anatomically inaccurate. You know, it is not, you know, it isn't below the soles of my feet, but it's represented in the spinal cord in that way, with the genitalia represented below the spinal cord. And that, in fact, continues all the way through the sensory pathways up to the brain, where your genitalia are represented in a sensory fashion below the soles of your feet. So the order which appears in the peripheral nervous system is maintained all the way through the central nervous system as well. Again, that's the kind of organising principle of how brains and central nervous systems are put together. Okay? Right, so I'm just going to tell you a little bit about the autonomic nervous system by way of a prelude to what's coming next. And what we're going to discover is that there are actually two parts to the autonomic nervous system, two divisions. One is called the sympathetic, and the other is called the parasympathetic divisions of the autonomic nervous system. Those are kind of their functional names, but they also have anatomical names which match. So the sympathetic nervous system is also called the thoracolumbar division, and the parasympathetic is also called the craniosacral division. Those are kind of their anatomical names. And the reason that they're given these anatomical names is because the sympathetic nervous system originates from motor neurons in the lateral horn in the thoraco and lumbar divisions of the spinal cord. In fact, from T1 all the way down to T12 and into the first part of the lumbar cord, L1 and L2, whereas the parasympathetic nervous system originates from motor neurons mainly in the brain, which are inside your skull or your cranium, with just a few from lateral horn motor neurons at the lowest sacral levels, S2 to S4. So again, this means there's a slight difference in the anatomical organisation and the functional organisation of different parts of the spinal cord, because there are no lateral horns in your cervical cord. Okay, there are no autonomic motor neurons that sit in your cervical cord, and therefore no lateral horn. Similarly, from L3 down to S1, there are no autonomic motor neurons, so there is no lateral horn at those levels. This is what the overall organisation of the autonomic nervous system looks like. Over here on the left in blue, we have the sympathetic or thoracic lumbar division, and over here in green, we have the parasympathetic or craniosacral division. I'm showing you this picture mainly to scare you, because the subject of the next lecture is to try to work our way through this. I just wanted to point out, for the time being, you see that the motor neurons in the lateral horn that give rise to the sympathetic nervous system are found all the way down the thoracic cord and in the top part of the lumbar cord. That's what these orange dots are meant to be. And there are axons coming out and eventually innervating all these tissues of your body, you know, your heart, your lungs, your heart, your liver, your stomach, digestive system, urinary system, genitalia. But actually, importantly, and this is why the autonomic nervous system is important for optometry, there's also a component that innervates a muscle inside your eye. This is the parasympathetic craniosacral division. You can see there's a bunch of nerves coming out from parts of the brain here, specifically from the lower part that I showed you before, called the brainstem. And there are four cranial nerves that give rise to parasympathetic autonomic outflow from your brain. And you can see that some of them also go to the heart and the lungs and the digestive system and to your urinary system and to your genitalia. The sacral bit of the cord is the one that supplies the very bottom part of your body, your rectum and your bladder and your naughty bits. But notice there's parasympathetic outflow to the eye. In fact, there are two muscles inside your eye which are innervated by the parasympathetic nervous system. And that's one of the reasons that the autonomics are so important. And we'll talk about those eye muscles in the next lecture. Okay? Right. Just to finish off a little bit about the typical spinal cord segment. Again, this is dorsal or posterior at the back, ventral and anterior at the front. This is about the white matter, which contains the myelinated axons that I mentioned before. Some of those axons are sensory, so they've come from spinal cord sensory neurons and are sending information or signals up to the brain. And some of them are motor. They've come from the brain and are going down to innervate the neurons of the ventral or lateral horn. And those axons run in bundles of axons found here in the white matter, which themselves have separate names. These regions are referred to as a funiculus. You've heard of a funicular railway, have you? Have you never been on a funicular railway? First, you should, anytime you get the chance, just jump on a funicular railway. When was the last time I was on one? Naples. Yeah. Naples has a bay. This is an aside, okay? Naples has a bay, and in October I was in Naples on holiday, and it was warm. I swam in the Bay of Naples. And much of the city, especially the poor bits, are on top of a hill. And to get from the top of the hill down towards the bay, where the central part of Naples is located, there's a funicular railway. It's a carriage that runs up a monorail, a little, you know, to the top, and runs down again. And the rail is completely fixed, but it's kind of at this angle. Okay, it's called a funicular railway. So it's like a track, and that's exactly why it's called a funiculus. It's a track. Okay. So there's a dorsal funiculus at the back, a ventral funiculus at the front, and a lateral funiculus on the side. And those funiculi, as we'll see, contain different axon pathways. Right, in brief, there are several somatic sensory pathways. All of these pathways originate in sensory receptors out in our body tissues who belong to axons that belong to dorsal root ganglion cells that send information into the spinal cord, specifically into the dorsal horn. And then from the dorsal horn, mainly, axons arise that travel up the spinal cord, particularly in the dorsal funiculus or in parts of the ventral and lateral funiculus over here. So there are several sensory pathways that run up the spinal cord to the brain. And those different pathways have different functions. They are specialized for different roles in sensation. Okay? These are anatomically distinct parallel pathways which have different functions. And this is an important concept or principle about how your brain is organized. Next year, as we will see, your visual system is based on exactly the same principles. There are several parallel pathways which are functionally specialized to do different jobs that run from your eye to your brain. And the same thing applies to sensory pathways in the spinal cord. There's one or a couple of particular pathways which are involved in somatic sensation, in conscious appreciation of touch or pain or thermal or proprioceptive sense. So there are pathways associated with conscious perception of the somatic sense. There are some that are involved in arousing the brain, alerting the brain to the fact that pain is stimuli are present, that tissue damage is in progress. And when the brain gets alerted to that, you might want to do something about it. But those pathways are distinct from these perceptual pathways. They're about brain arousal. And finally, there's a bunch of pathways which are actually involved in the control of our voluntary movements. You need to know, for example, where your limbs are in space. If you want to, use your hand to do something, to pick something up, you need to know where your hand is in space in order to move it to the location you want to go to. So you need to know where your body parts are in space in order to control your voluntary movements. And some of the sensory pathways that will run up the spinal cord have that specific job. The same principle applies to motor pathways that run in the spinal cord. These originate from what are known as upper, this is a jargon term, upper motor neurons located in your brain. And those upper motor neurons send axons down the spinal cord, so they're descending axon pathways that run in parallel, either in the lateral or ventral funiculi, to make connections with lower motor neurons in the ventral horn of the spinal cord that directly innervate skeletal muscles. And in fact, there are at least 3 sets of parallel pathways that come from your brain to these motor neurons in the ventral part of your cord. Some of those pathways are involved in the very fine control of really quite complex movements. For example, actions of your hand for grasping objects or playing the piano. Others are more concerned with maintaining your balance or posture, which is a much more kind of rudimentary thing, just making sure, and that is mediated by input from your vestibular system, you see? Your vestibular system tells you where your head is in space. I know my head is too far over to the side, so I better make an extensor movement to stop myself from falling over. Completely different pathway to those controlling fine, complex movements. And finally, there are pathways which are concerned more with regulating rhythmic, coordinated movements as you do when you're walking around. That's gait or locomotion. See that? That's it. That's flex. That's extend. Are you ready? Flex, extend, flex, extend, flex, extend, flex, extend, flex, extend. That's what we do when we walk. Contract, flexors, inhibit extensors. Contract, extensors, inhibit flexors. And then you can walk properly. And if you can't do that in sequence, then you fall over. And there are separate motor pathways that run down the spinal cord to ensure that you can do that. So, it's a kind of general principle for both sensory and motor systems that you have these separate pathways doing completely different jobs. Right. In fact, just to finish off, there are 2 major classes of motor neuron associated with the voluntary skeletal muscles. One of those classes is a large motor neuron, it's called alpha, and another class of motor neuron is a smaller one, and it's called gamma. And the large alpha motor neurons directly innervate the extrafusal muscle fibers, which when they contract make your muscles go short, in other words the whole muscle contracts. And the smaller gamma motor neurons innervate the intrafusal muscles where the muscle spindles are. You remember the muscle spindles are about the sensory system that tells you about the state of your muscle contraction. And these motor neurons, both the large and the small, are grouped together in what are known as motor neuron pools. Motor neuron pools. One pool of motor neurons innervates just one muscle. It's a general principle. Okay? They're all grouped together in the spinal cord, and that group of motor neurons, it could be 1,000 of them, only innervate one particular muscle in your body. That's their job. And there's a general mapping, topography to those motor neurons. The pools that are close to the midline of the spinal cord, is there a picture anywhere I can use? So like these guys here, see these are close to the midline of the spinal cord. They're medial, which is near the midline. They innervate medial muscles, you know, in the middle of your body. And these are lateral, towards the edge of the spinal cord. And they innervate muscles at the edge of your body, like the muscles in your hand. Okay? So medial motor neurons do the medial part of your body. Lateral motor neurons do the more lateral parts of your body. And it's these lateral parts of the ventral horn which are particularly big in your cervical and lumbar enlargements, because that's where your hands and your feet are. And this is just a little cartoon that shows the sort of general principle. This is a bunch of alpha motor neurons all stuck together here, these green ones. These are the cell bodies of those motor neurons, all together in a pool, and they're all sending their axons out down the ventral root through a spinal nerve, and look, lo and behold, each of those axons is innervating this particular muscle fibre belonging to a particular muscle. Right, so that's it for the lecture. Now it's a quiz time, which I hope you will engage with. I mean, you're not going to have to worry too much about this kind of thing when you grow up and become proper optometrists. But, you know, just to put into context, kind of clinically, some of the things that I've been saying. So look, this is a picture of the spinal cord that I showed you before, with its 29 different segments from cervical down to sacral. And what I'm going to ask you now is a little question. Say you see a person, because hopefully it won't be you, who's been unlucky enough to have damage to their spinal cord at this really low level here, which has chopped through their spinal cord completely. That's called a transection. And so this very low part here, S4, right at the very bottom, is now separated from the rest of the central nervous system. So they've got a transection of the spinal cord here at a very low sacral level, S3. What are their problems? They're going to have some problems. What sort of problems do you think they're going to have? Huh? They can't feel their feet. That is correct. Because the signals from the sensory receptors in their feet are going to travel up to the spinal cord to try to activate neurons here in S4 down towards the bottom. But those neurons here won't be able to send signals up the spinal cord to the brain any longer, so they'll lose sensation from their feet. What else will happen to their feet? Will they be able to move their feet? No. Because, see, the motor signals from the brain that are coming down to the motor neurons here in the ventral horn of the sacral cord that would normally get the muscles in the feet to contract, those signals stop here, they don't get to the motor neurons, so the feet are paralysed. And it would be on both sides, because this is a complete transection of the spinal cord. What else would they have? That's the sensory motor bits. Any guesses?
Pleasure.
Huh? Go for it.
Pleasure.
Well, they will lose a certain amount of pleasure,
Yeah.
because the sensory information from the genitalia won't be reaching the brain either, and something even perhaps even worse, depending on your age and your autonomics. Huh? Bladder control. Absolutely. They'll lose control of their bladder. So they'll be incontinent for wee-wee. Because the parasympathetic nervous system innervates the bladder, and causes it to contract, and that's gone. Anything else? They'll be incontinent for feces too, because they'll lose control of their rectum. And therefore they won't be able to contract the sphincter muscle of their anal canal until they'll poop themselves. So there you go, they'll lose sensation from the feet, their feet will be paralysed, and they'll be doubly incontinent. What about a lesion at T10? Of course they'll still be doubly incontinent because of similar reasons. What else has happened now? Now you're in the what do I say, the lower end of the thoracic cord. No legs, yeah. Actually, their legs are gone, okay? So they have no sensation from their legs. They won't be able to move the muscles in their legs. So basically they'll be in a wheelchair, okay? Because they won't be able to walk or maintain an erect posture, trying to stand up. What about T2, a high thoracic level up here? Actually, the main thing that is different between T2 and T10 from the point of view of the patient's perspective, and you as optometrists need to think about the patient's perspective, is that they will lose the ability to control their postural muscles in their trunk and their abdomen, so they won't be able to sit up in the wheelchair. Okay? They'll slouch because they can't maintain a posture sitting up. That's a real pain. What about T2? C2, the top one. This is basically, C stands for catastrophe, I think. It's really, really bad. Because now the level is here in the neck, and they're quadriplegic now. Now they can't feel or move their upper limbs either. And also, will they have trouble breathing? There is a particular bunch of nerves that innervate the lungs and help with breathing. They're called the phrenic nerves, and they come from C3 to C5. So there will be problems breathing too. So that's a real catastrophe. Okay, that's it. You have time. You have the opportunity to pause for a while, and then we'll finish off with the autonomic nervous system, which, as I said, is worth sticking around for. Okay then, ladies and gents, let's begin. So this last lecture of what I guess has been quite a long day for you is on the autonomic nervous system. It's also kind of our involuntary nervous system. It's also our unconscious nervous system. It's a bit of nervous system which is doing stuff behind the scenes of which we're not actually normally aware. The plan for the lecture is to spend maybe the first half looking at the functional organisation of the system, and then the second half looking at the kind of interesting bits, which are specifically those related to the muscles of the eye, and therefore of specific relevance to optometric practice. So I just wanted to begin by saying that I arrived here in the Department of, or Division of Optometry at City University in the year 2000, which presumably was before most of you were born. And when I arrived here, Ron Douglas, who you know and love, had already been here for over a decade. And on the very first day that I met him in the division, he said to me, Simon, great to see you, you're a neuroscientist, so you can take over my lectures on the autonomic nervous system. And I have to say, yes, because I was a new boy, but I found this quite depressing. It made me quite sad, because I thought, you know, the autonomic nervous system is the unconscious bit. It's not really doing anything very interesting. It's really quite boring, actually. And wow, is it complicated. You remember the picture I showed you in the first lecture to scare you? Whoa, I had that to confront it with. But actually, over the years, my attitude towards the autonomic nervous system has changed completely. I now think that it's an unsung hero doing really vital work behind the scenes that we simply can't do without, and it gets absolutely no credit for it at all. News. So that's what the lecture is going to be about. Now, because Ron used to give this lecture, he also wrote out a handout which I've made available to you on Moodle. If you flick to the very last page, you'll see Ron Douglas, January 1999, with a little bit from Simon Grant in January 2004. I think it's quite useful, but because it is quite a complicated system, and the handout existed, I decided, you know, to make it available to you too. Although this was written in 1999, don't panic. The autonomic nervous system hasn't really changed at all this century. Still pretty much the same. There are some bits in the handout that I've decided I don't want to talk to you about, because actually they are boring. So there's a bit here on page 3, which is all about Raymire and things. I just don't really find that very interesting, so I'm not going to tell you about that. There was something at the very end about, I'm not even sure I could pronounce it actually, achalasia, which apparently is a general term meaning failure of the autonomic fibres to relax smooth muscles. I'm not going to tell you about that. Which means they won't be in the exam, okay? So only the things I tell you about in the lecture and that are also in the handout are examinable, okay? If they're in the handout but not the lecture, they're not. Okay, so that was to kind of set the scene for what is to come. Right, so once again, you know, the two nervous systems, central versus peripheral, that there are sensory and motor components to the peripheral system, and as we've seen also within the central nervous system as well, and that the motor system has a voluntary component which is associated with innervating the voluntary skeletal muscles, and the involuntary or autonomic component is for smooth muscles and for glands, and as we'll see, for the organs inside our body. As it turns out, I mentioned in the previous lecture, the autonomic nervous system itself has two separate divisions to it, and they have functional names which are called sympathetic and parasympathetic. In general terms, the function of the sympathetic nervous system can be summarized by the terms fright, flight, or fight, the 3 Fs, okay? And the 3 Fs are all about running away or fighting, and all about energy expenditure. So that's the general function of the sympathetic nervous system, to mobilize your body for energy expenditure, to get you moving, to get you going.
The parasympathetic nervous system has completely the opposite effect. It's all about resting and digesting and recuperation and recovery. So that's the parasympathetic nervous system. It's for resting and digesting. And the two systems, normally operate whilst in opposition to each other to maintain, as we'll see, a kind of broad homeostatic balance of our body's energy sources, relating to the fact that the flight or fight system, otherwise known as a thoracolumbar, that's sympathetic, is associated with action and energy expenditure, recovered by the parasympathetic nervous system, also known as the craniosacral division, which is more about restoring the depleted energy stores, to replenish and recover. Right, just an interesting bit about the autonomic nervous system, okay? Just to prove that the autonomic nervous system really is an unsung hero, doing vital stuff in our bodies. The picture, these rather beautiful looking things that you see here, are almost transparent, but slightly blue or purple, are an organism called a sea squirt. And what sea squirts do when they're really young is they swim through the sea a bit, and then they find a rock somewhere, and then they anchor themselves to that rock, and they cement themselves onto that rock, and they're going to remain there for the rest of their lives. And all they're going to do is suck in seawater, which has got microorganisms in it, eat those microorganisms, and then excrete the stuff that they don't want. So one of the very first things that sea squirts do when they anchor themselves to the rock is to digest their own brains, because brains contain nutrients. But they keep some part of their nervous system. Specifically, they keep the autonomic parts, because the autonomic parts of the nervous system they need for energy use and for recovery. So they only have an autonomic nervous system once they get stuck on the rock. They ain't going anywhere. They're not moving. They don't care about anything. They just go, suck it in and blow it out. Right. So these are the sort of general functions, first of all associated with activation of the sympathetic nervous system, this column here in the middle. And as I've said, the sympathetic nervous system is for energy expenditure. And when you want to expend energy, all of these things happen at once. So this is a really mass action which is mediated by your sympathetic fight or flight system. So first of all, one of the things that you need to do is to increase your cardiac output. So your heart muscle increases its contraction. The blood flow to your heart needs to go up to support that increased cardiac output. So the coronary blood vessels dilate to allow more blood to flow to your heart. You breathe more often. Your respiration rate goes up as you're expending all this energy. Your bronchioles dilate, your trachea dilates. Your liver releases glucose from hepatocytes into your bloodstream, which travels to your brain and helps your brain be active. It also travels to the muscles, your skeletal muscles, and helps them contract, because the blood flow to your skeletal muscles is going to go up as well to get your, so your muscles can contract. Your sweat glands, they start to bung out loads of sweat because you're overheating, okay? All of these things go up or are upregulated by different parts of the sympathetic nervous system to expend that energy. But there are some things that, that could go on that you don't want to happen when you're expending energy. One of the things you don't want to do is to digest the meal that you've recently had, and you don't want to stop to take a wee-wee. So your digestive system and your urinary system is inhibited or shut down by the sympathetic nervous system. So these things are all excited, but these things are inhibited. Your salivary glands dry up and you don't secrete. The muscles, the smooth muscle in your digestive system stops contracting, so you can't move food around. Your kidneys reduce their urine output. Your bladder remains completely contracted so that it won't release urine, okay? And so you get some things that are upregulated by sympathetic mass action and some things that are inhibited. As it happens, as we'll see later, the sympathetic nervous system also directly or innervates a muscle inside your iris, and it's called the dilator muscle. Shock horror, this means your pupil dilates when your sympathetic nervous system is active. Once all that energy expenditure is over, you need to kind of stop and recover, and that's when the parasympathetic nervous system kicks in to try to maintain the balance. You know, this is a thing which is known as homeostasis. So everything now happens in reverse. When your parasympathetic nervous system is working, there's another mass action, but now the contraction of your heart muscle decreases. Okay, so you're resting. The blood flow through the coronary arteries that supply the heart muscle, that decreases. Your respiration rate reduces. These things also happen in the sense that your sweat glands also stop secreting. That's not because the parasympathetic nervous system innervates the sweat glands, it's because there is an absence of sympathetic innervation to the sweat glands. So just the absence of sympathetic allows the sweat glands to slow down. Similarly for the liver. But these two things are definite inhibitions of your heart rate and your respiration rate. But now also you need to recover, so you've got to excite or upregulate your digestive system. Now is the opportunity to, digest and absorb that food that you ate in the last meal. Now's the time to take a stop at a toilet and do a wee-wee. So your digestive system and your urinary system now, increase their activity. As it happens, the parasympathetic nervous system also innervates two other muscles inside your eye. Specifically, it innervates the iris sphincter muscle, which is responsible for constricting your pupil, i.e. the opposite to dilation, and it also innervates a muscle called the ciliary muscle that contracts that muscle and causes the lens inside your eye to become rounder. That's for accommodation and for focusing at near. And we'll talk a little bit more about the iris dilators and sphincters at the end of the lecture. Okay, so for now I just want to talk about some major differences. Some major differences between the somatic, voluntary part of the motor system and the two divisions of the autonomic motor system, and also a little bit about differences between sympathetic and parasympathetic, but at the cellular rather than gross anatomical level. So what I'm going to do is look at the location of the different motor neurons in the central nervous system. We're going to look at the fact that there's a synapse in the autonomic pathway, which is mediated via things which are called autonomic ganglion cells. We'll talk about different axon lengths and their degree of myelination, because this has significance for conduction velocity. And we'll also talk about the neurotransmitters that the systems use and about their receptors. I've already mentioned, that the location within the central nervous system of the motor neurons that are responsible for innervation of skeletal muscles via the somatic motor system, and for other things via the autonomic nervous system is different. For example, I mentioned, in the last lecture, in the ventral horn of the spinal cord you have neurons that go to skeletal muscles, and in the lateral horn you have the autonomic motor neurons. So they're in different locations, and they're doing different jobs. So, excuse me, too much talking. And in fact, as we'll discover subsequently, i.e. next week, the brainstem also has groups of neurons that are somatic. They innervate the skeletal muscles in your face, and it also has autonomic motor neurons that innervate ganglia, which are responsible for innervating the smooth muscles or glands in your face. Okay, so this kind of little cartoon then is meant to help me talk to you about those various things that I mentioned in the previous panel. So this bar here is meant to be the central nervous system. For the sake of argument, it's the spinal cord. And all this stuff out here is the peripheral nervous system containing nerves and axons. And here are the various target tissues that the motor system innervates. So this one here, this blob, is meant to be the cell body of a somatic motor neuron inside the spinal cord. Here's its axon in the peripheral nervous system, directly innervating a skeletal muscle cell. These are the cell bodies of sympathetic and parasympathetic motor neurons in the spinal cord. These are their axons coming out, and that eventually these two systems will innervate either smooth muscles, which, as you know, are involuntary muscles, cardiac muscles, which is basically heart muscle, or various glands or organs. So the first thing to point out is that the somatic motor system is what is known as a monosynaptic pathway. In other words, the motor neurons in the spinal cord have axons that come out, this is the somatic motor fibre, and directly innervate skeletal muscles with one synapse. That's monosynaptic. But the autonomic nervous system is disynaptic. What does this mean? It means that the autonomic motor neurons of both the sympathetic and parasympathetic branches have axons that come out and make connection with neurons in the peripheral nervous system, which are collectively known as autonomic ganglion cells. And those synapses activate the autonomic ganglion cells, but then have axons of their own, which travel to the particular smooth muscle or cardiac muscle or gland that they're going to innervate. So that's a disynaptic pathway. There are two synapses involved in the autonomic nervous system. From the motor neurons in the CNS, what are known as preganglionic axons or fibres come out and innervate autonomic ganglion cells, which are grouped together in knots in different locations around our bodies. And those autonomic ganglion cells have axons of their own, which are referred to as postganglionic axons or fibres, which then directly actually innervate the target tissue. So what this means, first of all, is that the somatic voluntary motor system is faster than the autonomic, because there's always a delay in processing signals at a synapse. So the voluntary motor system is faster than the autonomic. And in fact, there's an even better reason why the voluntary motor is faster than the autonomic. And it's this. The diameter of the axons that belong to somatic motor neurons are generally really quite wide, and those axons have lots and lots of myelin around them. What does a wide diameter axon with lots of myelin mean? It means a very fast impulse conduction. And so the skeletal motor system has fast impulse conduction. In the autonomic nervous system, only the axons of the preganglionic fibres from here and here have any myelin at all, and those axons are also quite small in diameter. These axons of the postganglionic fibres have no myelin at all. They're completely unmyelinated. And of course, and they're very thin. Thin axons, small diameter, no myelin at all, means slow impulse conduction. So overall, the somatic motor system to voluntary skeletal muscles is fast, and the autonomic motor system to these other effector tissues is much, much slower. And there's a really good reason for that. You know, if you're walking along and you trip, you know, you've got to do something voluntarily to stop yourself from falling over. You can't go, ooh, I'm falling over. Maybe I'll think about this for a bit before, and then you use the face to break your fall. It's not, it doesn't work like that. The voluntary motor system has got to be quick. The autonomic nervous system can take a bit more time. We're only talking about maybe a second to do stuff, maybe even less than that. What else did I want to tell you? Oh yeah, there's something else in the picture which is true, and that is the sympathetic branch. You see, the initial preganglionic fibre is quite short, and the ganglion cells seem to be quite close to the central nervous system, and so the postganglionic fibres seem to be quite long to innervate their target cells, but the converse is true of the parasympathetic. The parasympathetic motor neurons in the central nervous system seem to have long axons which go to a ganglion which is quite close to the target tissue, and that is true. In fact, the ganglia of the parasympathetic branch are kind of known collectively as the terminal ganglia, because they tend to be found very, very close to their target, and sometimes actually in amongst their target tissues. Here's an example. the autonomic ganglion cells of the parasympathetic nervous system that innervate the muscles of your bladder are in the wall of your bladder, right next to the muscles they supply. So this axon here is really short, but the autonomic ganglia that innervates your bladder sympathetically is a long way away from your bladder. So this one is quite long. And actually, as we'll see, the same thing applies to the innervation of the muscles inside your eye. The sympathetic ganglia for eye muscles is terminal, close to your eyeball, whereas the sympathetic ganglia for the dilator muscle is quite a long way away. Okay? I think I covered everything in there that I wanted to. Yes. What I didn't do was the receptors, and so this is another really important functional distinction between somatic and the two branches of the autonomic. Have you heard of a neurotransmitter called acetylcholine? I think you have. I think Ron Douglas has told you about acetylcholine. Acetylcholine is usually an excitatory neurotransmitter. It is used by somatic motor neurons. They release this neurotransmitter onto skeletal muscle cells, and it causes those muscle cells to fire action potentials and to contract. The motor neurons of both the sympathetic, which is this one, and the parasympathetic nervous system also use acetylcholine as their neurotransmitter, and that neurotransmitter here is released by the parasympathetic nervous system onto the ganglion cells of the parasympathetic autonomic ganglia, and those ganglia also use acetylcholine as their neurotransmitter onto their target tissues. But that is not true for the sympathetic nervous system. The ganglion cells of the sympathetic nervous system receive input from motor neurons in the CNS, and that neurotransmitter is acetylcholine, but they release a different neurotransmitter onto their target tissues. That neurotransmitter is called noradrenaline. In North America, it's called norepinephrine, but they're two names for the same thing. Okay? So in the UK, it's noradrenaline, which is the transmitter released by the sympathetic branch onto the target, but acetylcholine from the sympathetic branch onto the target. So pretty much everything is acetylcholine apart from this sympathetic ganglion to terminal input, which is noradrenaline. Okay, so two completely neurotransmitters released onto the target tissues by the two branches of the ANS. Okay, so that was the cellular stuff. Now back a little bit to the kind of gross. And I just wanted to look first of all at the right-hand side, the kind of gross organisation of the parasympathetic or craniosacral division. It's called craniosacral because there are loads and loads of motor neurons up here in the brainstem, which is part of your brain inside the cranium, that give rise to parasympathetic outflow, and just a little bit down here at the very bottom of your sacral cord that also give rise to parasympathetic outflow, hence craniosacral. Note the hidden map. What do I mean by this? As we're just about to see, there are 4 cranial nerves that originate from motor neurons in the brainstem that give rise to parasympathetic outflow that innervate different tissues either in your head or in your body. The top one of those nerves is here, okay, where the arrow is, and you can see the axons are coming out and eventually are going to innervate a muscle, or muscles actually, inside your eye, which is quite high up in your body. There's another nerve that comes out of the brainstem about halfway down, you see, and that one goes across here, and it innervates the salivary gland, which is down here, below your eye. See, so we're moving down the brain, and we're moving down the body. This one here is a huge nerve, and its axons come out, and in fact, it comes from the brain, but it doesn't innervate things in the face. The axons travel down into the whole of your trunk and abdomen and innervate your heart, your lungs, your liver, your digestive system, apart from the few parasympathetic nerves that come out of the sacral cord that do your rectum and your bladder and your naughty bits. Okay? So there is a kind of top-down map to the nerves coming out of your central nervous system. Top ones go to the eye, intermediate to salivary glands, lower down to these organs, and lowest of all to the lowest parts of your body. We're going to do this again next week when we come on to cranial nerves, but I'm just going to give you this as a prelude because these are the types of words you're going to have to remember, unfortunately, when we do cranial nerves. There are 12 pairs of cranial nerves, and 4 of them have parasympathetic outflow or output in them, okay? They're cranial nerves number 3, 7, 9, and 10, because the cranial nerves are numbered 1 to 12. The numbers also have names. 3 is oculomotor, 7 is facial, 9 is glossopharyngeal, and 10 is vagus. As I'll tell you next week, some of these names are a little bit of a clue as to what the nerves does. You see, oculomotor. Take a guess. Motor movement, yeah? Ocular eye, you bet. It's all about eye movements. Glossopharyngeal, sounds impossible. Glottis, pharynx, yeah, associated with that part of your body. Vagus, no chance. Too vague. Right, so here's a little bit of detail. The third oculomotor nerve has voluntary bits, which we'll talk about next week, but it also has an autonomic component. It arises from a nucleus in your brainstem. That nucleus has a name. You're going to encounter this name many times over the course of the next few years, and you need to remember it for the rest of your life. It is called the Edinger-Westphal nucleus, and it's inside your brainstem. That nucleus gives rise to axons, so those are the pre-ganglionic fibres that run down the third cranial nerve, and they terminate on autonomic ganglion cells in a thing called the ciliary ganglion. That ganglion is a terminal ganglion. It's just right behind your eyeball in your orbit, very close to the muscles it's going to innervate. The ciliary ganglion gives rise, those ganglion cells, to axons of their own, so those are the postganglionic fibres, and they travel inside your eyeball to innervate the iris sphincter muscle for pupillary constriction in response to light, and to the ciliary muscle for accommodation so your lens goes fat and you can see it near. And all that is really, really important for optometry. This is another one which is really important for optometry. The 7th cranial nerve is known as facial, mainly because it innervates the muscles of facial expression. But it has, which are voluntary, but it has autonomic components. One which goes via this ganglion, wonderful, the pterygopalatine. And it activates neurons in those ganglion cells that give rise to a nerve that innervates cells of the lacrimal gland. And the lacrimal gland is really important. It produces the watery component of our tears. And if the lacrimal gland can't produce the watery component of our tears, our corneas dry up. That's a condition called dry eye. And if they dry up too much, they start to split, they become eroded, and then they get infected, and then your cornea goes opaque, and then you go blind. Okay? So this innervation of the lacrimal gland is really quite important. It's the difference between you and blindness. There's also another component that goes through a different ganglion called the geniculate that supplies 2 of your salivary glands. Glossopharyngeal, okay, has nerves that go to another ganglion called otic that supplies your largest salivary gland, which is called the parotid, that actually does the watery component in your saliva. And this one here, number 10, the vagus, is huge. It's responsible for about 90% of all the parasympathetic axons that come out of your central nervous system, because it innervates loads of different ganglia supplying the heart, the liver, the respiratory system, digestive system, and parts of the urinary tract. Okay? Right. Now the sympathetic thoracolumbar division, which is the thing on the left, kind of in blue, on the blue background. So the motor neurons that give rise to the sympathetic branch are all within the spinal cord. There are none in the brain at all, okay? And here they are. from the top level of the thoracic cord all the way down to the bottom, so that's T1 to T10, and intruding into the first, second, possibly third lumbar divisions. These motor neurons in the spinal cord have axons that come out, so those are the pre-ganglionic fibres, and they make contact mainly with ganglion cells that are just outside the spinal cord, just outside the backbone, and run together alongside the spinal cord as this thing is called, the sympathetic chain of ganglia, or the paravertebral chain, because they're all kind of linked to each other. You can see there are swellings here, because there are 22 knots, K-N-O-T, of sympathetic ganglion cells just outside the spinal cord that these neurons inside the spinal cord innervate. Those sympathetic ganglion cells then give rise to axons of their own, which travel usually quite a long distance to innervate their target tissue. And as shown here in green, the synapses on the end of those postganglionic fibres use NE, norepinephrine, or noradrenaline as their neurotransmitter. Notice, I don't really want to tell you much more about the sympathetic chain of vertebral ganglia because it's really boring. It's in the handout, but forget it. All you need to know is that there are 3 of these ganglia outside the spinal cord, level with the cervical part of the cord and the rest are lower down. One of the reasons you need to know that is because the very top ganglion here, which I put an asterisk by, has a name. It's called the superior cervical ganglion. Superior because it's the uppermost, cervical because it's next to the cervical cord, and it's a group of ganglion cells. And look, the axons of those ganglion cells travel up and innervate a muscle inside the eye. So that's really important for optometry. Notice there's kind of a hidden map in here. You see the superior cervical ganglion also has other cells that inhibit salivation. Lower down, there are other ganglion cells below the cervical that supply input to the heart and the lungs. Further down are other ganglion cells that supply input to organs lower down in your abdomen. So again, there's a kind of top-down arrangement here within the sympathetic nervous system and the sympathetic chain. A couple of other points that are reasonably interesting. There's a big nerve called the splanchnic nerve, which actually is this one here, that actually travels a fairly long distance, and it innervates sympathetic ganglion cells in a knot, which are known collectively as the solar plexus, which is around about here, just underneath your diaphragm. If you get hit in the solar plexus, has that ever happened accidentally to any of you? You don't want that to happen, I can tell you. It's quite unpleasant to be hit in the solar plexus, because you find it really quite difficult to breathe. And also, there is one component of the sympathetic nervous system that completely bucks the trend, or one of the trends that I mentioned before, and that is this bit. Okay, so it comes from this lowest part of the thoracic cord, and the axons come out, and they don't go through a ganglion at all. They directly innervate their target tissue. So this pathway is monosynaptic. It's the only one anywhere in the autonomic nervous system, and it goes specifically to the adrenal medulla in the adrenal gland, which has cells that release adrenaline or epinephrine into your bloodstream, which is the humoral or hormonal component of the fight and flight system. So there are neurons inside the spinal cord that directly innervate cells of the adrenal medulla to release adrenaline, which completely boosts this 3F system so that you expend the energy in the way you want to. Right. So we're almost getting to the really exciting bit, but we're not there yet. Question 2 is where the exciting bit begins. Right. I want to pose 2 questions here and answer those questions. Here's the first question. How can the release of the same neurotransmitter, for example noradrenaline, cause excitation in some of its effectors, like the heart muscle, which is excited, and inhibition of others, like gastric glands or salivary glands. How can the same neurotransmitter cause excitation somewhere and inhibition somewhere else? And the answer is there are two types of receptor to the neurotransmitter noradrenaline. And again, this is a really important principle and concept about how the nervous system works. Second question, which is moving on to the interesting bit, how can the sympathetic and parasympathetic nervous systems have different effects on the iris or the pupil? How is it that the sympathetic nervous system causes dilation of the pupil, but the parasympathetic causes constriction? This isn't about different neurotransmitters solely now, it's about different muscles. Now let's look at the answer to question 1 first, which is the complex bit. Okay, so this is a little bit of noradrenaline receptor pharmacology. And in brief, there are two types of receptor that sit in the membranes of different effector cells. And they are different effector cells, not as shown in the picture here in the same cell membrane. That never happens. Otherwise it wouldn't know what the hell to do. So some cells have the excitatory type of receptor, which is called a beta receptor. So heart muscle, for example, would have beta receptors in their membranes. And other receptors have an inhibitory type, and they're called alpha or alpha-2 receptors. So your salivary glands, for example, would have the alpha-2, the inhibitory. And this is the principle of how the whole system operates. So here is, look, a package of noradrenaline or norepinephrine here, binding to this particular excitatory type of beta receptor situated here in the cell membrane of a thing that's going to get excited. That receptor isn't an ion channel, there's no hole in the middle of it, it's a G protein coupled receptor that activates proteins on the other side inside the cell, okay, down here. And the activation of those G proteins actually regulates the activity of another enzyme which is called adenyl cyclase. And when adenyl cyclase is activated by this excitatory beta receptor, what happens is that potassium ions inside the cell stop leaving. That's called potassium plus efflux is reduced. So there are potassium ions inside the cell and their channels close, so they don't go out any longer. So what happens? There is a build-up of positive charged potassium ions inside the cell, which makes the inside of the cell more positive. That's called depolarization, and therefore the cell becomes excited. This is the alpha-2 receptor. Look, here's a package of noradrenaline binding to this receptor. Again, it's not an ion channel with a hole in the middle. It's linked to another G protein on the inside of the cell, and when it's activated, it does something else to adenyl cyclase that changes the response of potassium channels also to their efflux. Now, however, the channels open and potassium efflux is increased. So potassium ions inside the cell leave in large numbers. Because those potassium ions are positively charged and they're going out, the inside of the cell becomes more negatively charged. That is called hyperpolarization, and it means inhibition. So these cells are inhibited. And they would be, you know, so, as I say, cells of the salivary glands or of your digestive system or in the walls of your bladder, because those are inhibited by sympathetic input. So that's how the nervous system can produce two effects by releasing exactly the same neurotransmitter, because they're different receptors. One excitatory, one inhibitory. As we'll see next year, same thing happens in your retina. Right, so now I just want to get on and talk about eyes, but also I think these terms you should be familiar with. I think Ron's already mentioned them to you. So this is about the receptor pharmacology now for acetylcholine receptors. And in fact, there are two classes of acetylcholine receptor in our bodies. They never sit together here in the same cell membrane. They're always in different cells. One is called a nicotinic receptor, and this is a conventional ion channel with a hole in the middle, and the other is called a muscarinic receptor. They're named, these two types of acetylcholine receptor, for naturally occurring substances that mimic the effect of acetylcholine. So, those substances are known as agonists. So an agonist mimics the effect of the release of the neurotransmitter inside your body. You were familiar with that term, yeah? You'd heard of agonist? Maybe. So the agonist of the nicotinic receptor is nicotine. It's the substance that you find in tobacco leaves. And nicotine binds to the nicotinic receptors and causes the channel to open and the cell becomes excited. And nicotinic receptors are the ones you have in all your skeletal muscles. Okay? In your smooth muscles and glands, however, you have this other type of receptor which is called muscarinic. This is named after a material that you find in many species of mushroom, and it's called muscarine. And muscarine binds to these receptors and will have one of two effects, because there are different classes of muscarinic receptor. In fact, there are 5 known muscarinic receptors. Muscarinic receptors 1 and 5 are excitatory, so when acetylcholine binds to those receptors, the cell is excited, and this is the type of receptor you find in the parasympathetic nervous system. So these would be the ones that you have in your iris sphincter muscle. And in other tissues, there are other muscarinic receptors. They're M2 and M4, which when muscarine binds to them, the cell is inhibited. So it's the same principle as we saw before for the noradrenaline. Okay? So in the autonomic nervous system, for the parasympathetics, it activates muscarinic receptors, which are either excitatory or inhibitory in character. And those receptors also have things which antagonize or prevent their action. And those things are called antagonists. A classic antagonist of the nicotinic receptor is a material called curare. Curare is a paralyzing agent. It's used in conventional medicine to paralyze patients when you need to. You would then have to artificially ventilate them. But I can tell you where curare comes from. It comes from plants in South America. And refined forms of curare in South America are used by native South Americans to dip the arrows that they use in blowpipes into. You know, the native South Americans have blowpipes. They're really long tubes, and they put little arrows inside them with feathers on the end and a tip, and they dip the tip in curare, and they go, and that arrow flies through the air and hits an animal, like a pig or something, in the forest, and the curare gets into the bloodstream of the pig, and it goes to all the nicotinic receptors, including in their diaphragm muscles, and they suffocate and die, and then the Indians eat them. Okay? So that's where curare comes from. It's an antagonist of the nicotinic receptors. One of the classic antagonists of the muscarinic receptor is this material. Ever heard of it? It's called atropine, okay? And it's an antagonist of muscarinic receptors. Right, so now we've got to the iris muscles. And we're going to do, Ron's going to tell you loads more about the iris muscles, or other people will, later on this term. But this is just a kind of cross-section through this part of the eye, this kind of oval elliptical structure here, that's the lens, and this region is the region of the corneal iridial junction. And what it's showing here is this is the iris, okay, in cross-section. This is the coloured bit of your eye. And what it's showing is that there is quite a long muscle. It starts here at the base of the iris, and it travels quite a long way up, in fact about 2/3 of the length up the iris, and that muscle is called the dilator muscle. And then running circumferentially around the edge of the iris is the iris sphincter muscle. And this is trying to depict these two muscles kind of head on. So look, this black space in the middle is your pupil. This is the coloured bit of the iris. This happens to be a person who's got brown eyes. And here, inside the brown of the iris, is the orientation of the muscle cells of the dilator and sphincter. So the blue radially oriented muscles, like this, coming out almost like the spokes of a wheel. Those are the pupil dilator muscles. Okay? When they contract, their length gets shorter, and so the round circle in the space opens up. You see? So that's called dilation. And that's the job of the dilator muscles, is to dilate your pupil under circumstances of low illumination, for example, when you want to get more light in. This is the orientation. It's completely different in red of the sphincter muscle. Like all sphincters, it's a circular muscle that rounds around the very edge, the rim of your pupil, see? So when those muscle cells contract, they get shorter, and so the space in the middle gets smaller. Okay? So that's pupil constriction. So the radials dilate and the circulars constrict. Simple as. And as I've told you several times already, the dilator muscle gets input from the sympathetic branch of the nervous system via neurons in the superior cervical ganglion, and the sphincter muscle gets parasympathetic innervation by way of the ciliary ganglion. And here are some important antagonists and agonists associated with the two neurotransmitter systems. Look, over here on the right are some agonists of the sympathetic nervous system. These are all classics. If you ingest these in any way, then your pupil will dilate. Here are two real classics: amphetamine and cocaine. And I can tell you from personal experience, if you ingest either of those, your pupils will dilate. This is the classic antagonist of the beta type of receptor for noradrenaline. It's called propanol. It's the thing that, it's called a beta blocker, you know, it's a classic antagonist. One of the other things that you can do to manipulate neurotransmitter systems is to think about the enzymes that are present in synaptic clefts, which degrade the neurotransmitter once it's been used. So once you release neurotransmitter and it binds to the receptor, there's still some of the transmitter floating around, and you don't want that to happen. So there are enzymes which are resident in the synapse that will break the neurotransmitter down and render it inactive. And those are known as neurotransmitter degradative enzymes, and there are blockers of those enzymes. In other words, drugs that will keep the neurotransmitter around in the synapse when it should normally be broken down, and that is basically to boost the transmitter system. And here's a classic neurotransmitter degradative enzyme blocker of the noradrenaline system, and it's called a monoamine oxidase inhibitor. So, quite often in optometric practice, you want to dilate the pupil, don't you, of a patient that you want to look at. And there are various reasons why you'd want to do that. One is to, you know, sort of get your ophthalmoscope in to have a good look around in the back of their eye. And it really helps if their pupil's dilated, to give you access to do that. And also you want to do it sometimes for cycloplegic refraction of the patient, to get a really accurate measure of just what their refractive error is. And under those circumstances, it's also quite useful to dilate their pupil. And so, I mean, an obvious way of dilating their pupil would be to apply some sort of agonist of the sympathetic nervous system directly onto the surface of their cornea. That agonist will then travel into their eye. It will activate the dilator muscle, and bingo, their pupil will dilate. But look, the agonists of the noradrenaline system are these Class A or Class B drugs. I mean, they're illegal. You can't drop cocaine on the eyeball of a young child to do a cycloplegic refraction, unless you have permission from a pharmacologist or from a pharmacist to do that. But it's actually really quite rare to do that. So you can't really dilate the pupil by using the agonist. An alternative would be to increase the availability of the neurotransmitter onto the dilator muscle. So you could use a neurotransmitter degradative enzyme blocker to do that, and that would be monoamine oxidase inhibitor. Unfortunately, you can't, because these drugs are psychoactive when they're actually used in the treatment of quite serious mental conditions like schizophrenia. So you can't really manipulate the noradrenergic system in order to produce pupil dilation. So what you actually do is to manipulate the parasympathetic system. And what you do is to drop onto the eyeball antagonists of muscarinic receptors on the iris sphincter muscle. Why would you do that? Because when you drop an antagonist of the muscarinic receptor onto that receptor, it blocks the receptor, and therefore the sphincter muscle can't constrict the pupil any longer. But the dilator muscle is still working. It's unopposed, and therefore the pupil dilates. So you get dilation of the pupil by blocking the constrictor, not by activating the dilator. And indeed, that's what you do. Atropine is a fairly common drug to produce pupil dilation. I'm told by optometrists that I can never say this. Anyone want to shout it out? Thank you. Tropicamide is the drug of choice. It's a muscarinic blocker used in adults, and cyclopentate is the one that you tend to use in kids, because this paralyzes their ciliary muscle, which means they can't accommodate while you're trying to do the refraction. You know, if you say to an adult subject, could you please focus on this thing while I look at your refraction? And most adults will actually do what you ask them to. But if you take a 5-year-old child and say, could you please focus on this thing while I look at your refraction, do you think they're going to do it? No, they're not. They ain't. So you paralyze their accommodation so they can't do anything else. Okay? And this drug, cyclopentate, works on the ciliary muscle and blocks it, so that it blocks their ability to accommodate a near. Now sometimes, oh shit, it's getting late. Sometimes you have to try to constrict somebody's pupil because it seems to be permanently or pathologically dilated. And this happens. You'll sometimes see a patient, in a optometric practice, and it's often just one eye alone, where the pupil is really wide and the other one's the normal size. And you think, well, why the hell is this pupil dilated? And there are two possible reasons for it. One is the muscle. What we're talking about here is the constrictor muscle isn't working properly, is damaged in some way. That would be a myogenic reason for the dilation. Or it could be the nerve supply to the constrictor muscle isn't working, and therefore the pupil is dilated. And so in order to do the differential diagnosis here, the standard approach is to put on a blocker of the degradative enzyme of acetylcholine, which is acetylcholine esterase, to block the removal of acetylcholine, therefore increasing its availability at the nerve-muscle junction. And here are two drugs that do that, estrogen and physostigmine. And if you do that, you apply these blockers onto the eyeball, and the pupil now constricts. It means the muscle's working, because you've got the constriction. Okay? So therefore the problem, the pathology, is likely to be neurogenic. The neuro supply to the muscle isn't working, because the muscle is fine. If you drop these drugs on and the pupil doesn't constrict, even though you've boosted the supply of acetylcholine, it means the muscle isn't working. So the problem is therefore myogenic. So its genesis is in the muscle itself. So these are the opposite neurogenic pathologies. A dilated pupil is called mydriasis. Constricted pupil is called miosis, now spelt correctly, as opposed to incorrectly in your notes. And dilation of the pupil is associated with damage to the parasympathetic ganglion or nerve, which paralyzes the iris constrictor, and so the dilator is unopposed. And a classic condition associated with it is called Adie's tonic pupil. Miosis is associated with damage to the sympathetic ganglion or the nerve, which paralyzes the dilator, so the constrictor is unopposed, and this happens in Horn syndrome. This is Adie's pupil, Adie's tonic pupil in the left eye. See, look, the right eye's fine. That's the size the pupil ought to be, but the left eye is showing mydriasis. So here the problem is with parasympathetic innervation of the iris sphincter muscle. That innervation begins in the midbrain, and something may have happened to the third nerve, some sort of break, or maybe to the ciliary ganglion, or maybe to its post-ganglionic fibres that have prevented the iris sphincter muscle from being innervated, so the pupil can't constrict, and it dilates because the dilator is unopposed. This is the sympathetic innervation of the iris muscle. Here again, on the right, it's good. The pupils are right size, but here you can see it's abnormally constricted. This is meiosis. And also you might be able to see that the upper eyelid is a bit down. When the upper eyelid droops, that condition is called ptosis. So this patient has both a mild ptosis and meiosis of the left eye, and this is due to damage to the sympathetic innervation. But here it is. This is the superior cervical ganglion. Here are the postganglionic fibres. Some of them run inside the eye and innervate the dilator muscle, which isn't now working, so the pupil constricts. And some of those fibres run up and innervate a muscle. It's a smooth muscle in your upper eyelid, which is called Muller's, or the superior tarsal muscle, and its job is to help the eyelid stay open. So you've got sympathetic supply to a muscle in your eyelid that makes your eyelid stay open, and innervation to a dilator, which dilates your pupil. Does that sound sensible? It's really smart. If you're in a dim environment and you want to open your pupils to get more light in, it's a smart move to open your eyelids as well to help with that. But when those things aren't working, then your eyelid droops and you get ptosis and you get this meiosis of the pupil. You also get actually lack of facial sweating, which is called anhidrosis. And those three things are the classic signs of a condition called Horner's syndrome. Quick MCQ. Innervation of iris sphincter. 1. A. Sympathetic nervous system? No. B. Seventh cranial nerve? No. Third. Activates smooth muscle fibres circularly in the pupil margin? Yeah. Not just because it's the longest answer, but also true. And it's not damaged in Horner's, that's sympathetic. Cool, done. See you next week.